And now we have Dr. Ivaylo Iotchev. He joined our senior family project in 2016, and since then he has been studying the expression of sleep spindles in the brain of dogs using EEG. Today, he will share an overview of the results concerning what these brief bursts of neural activity can tell us about the cognitive capacity of young and old dogs. Please welcome Dr. Ivaylo Iotchev on the sleep spindle as a marker of the cognitive aging in dogs.
Good. One second. Good evening, everyone. I hope I'm audible and visible. Well, I will assume
yes.
so. Good. Well, as already was mentioned in the introduction, I'm one of the people here at the department who like to approach questions about the dog brain using electroencephalography, or EEG, which is a classical method measuring voltage changes across time caused by the brain's activity that we can pick up with surface electrodes from the scalp of humans and animals. What I will first show you here are three alternative representations of the same segment of EEG recorded from a sleeping dog, like this one here. Specifically, we're looking at non-REM sleep, which is dominated by large low-frequency waves, known as delta, and this is what we see in the uppermost representation under A, which is the raw, untouched signal. If we filter delta activity away, shorter bursts of higher frequency become visible. These are the sleep spindles that I will talk about, and you can see one in the middle of the middle figure under B. Why are sleep spindles interesting and important to us? Well, first of all, historic work in the cat, which was invasive and experimental, has revealed the underlying mechanisms of the sleep spindle, which I'm not going to talk about today, but this is important to note because it is not always the case that in surface EEG we know what is happening inside the brain. This is particularly true for some types of epileptic seizures, for example, where the pathology can be very unique to the patient. So to put it short, when we see a sleep spindle, we have an idea of what is happening in the brain. In addition to this, and this is more closely tied to the title of today's topic, several features of the sleep spindle have been shown repeatedly in humans to correlate with important cognitive capacities like sleep-dependent learning and intelligence. In particular, for today's talk, I will focus on our results relating to sleep spindle density and sleep spindle frequency in the context of studying cognitive aging. To explain these two features of the sleep spindle, I would like to use the following schematic figure that you see at the bottom of this slide. Here, under the red arrows, you see two typical sleep spindles, and the number of such events per minute of non-REM sleep is what we call sleep spindle density. This is a measure of sleep spindle occurrence. Not to be confused, sleep spindle frequency is not about the occurrence of sleep spindles. Rather, as we can also see in the figure, each spindle consists in itself of several small waves that follow each other in succession. And the number of waves per second that we see in each spindle is that spindle's frequency. It's not only important for the results which I will show you, but spindle frequency is a characteristic used to distinguish so-called slow and fast spindles in humans, dogs, and rats, where slow spindles are those who exhibit frequencies lower than 13 Hz, and fast spindles exhibit frequencies higher than 13 Hz. And importantly, that's not an arbitrary distinction either, where we just cut the frequency range of sleep spindles into two halves. No, it is the case that these two types show a different distribution over the scalp. So electrodes placed at the center or even the back of the head are those where we pick up most of the fast spindles, for example, although both types can be observed at each electrode. Now let me come to the work which has accumulated in the last 4.5 years with regards to cognition and aging. First of all, my colleague Dr. Iotchev was one of the driving forces behind the paradigm we use for studying sleep-dependent learning in the dog, although we did not initially use it to test sleep spindle effects. And I will try to describe the core idea, which is that we take dogs which already perform quite reliably on simple commands like sit and lay down, and because we're situated in Budapest, these dogs have been trained to respond to these commands in Hungarian. And so in our learning experiments, which by now are more than one, we teach these dogs to respond to the English equivalents of these commands. And so after training is complete, we test the dog's performance on the novel task before and after we send them to the sleeping laboratory where we collect their EEG signals. Now, because of this arrangement, we can look both at the recall performance after sleep, but also at the relative difference in performance before and after sleep on the novel task. And importantly, between 2017 and 2020, we already have observed that each of these measures of sleep-dependent learning are positively associated with the density of sleep spindles. That is, how many sleep spindles we see per minute of non-REM sleep. Also crucial, we first observed the effect in 2017, but since we do not use invasive methods and cannot directly look at the mechanisms which we suspect to underlie this phenomenon, we wanted to have more certainty, and we sought to see if we can replicate this observation in 2020 when we already had 3 similar data sets. And importantly, what we found is that if we averaged individual measurements of a single dog's learning performance and sleep spindles, because for some of these experiments we had several measurements of each, then observing this association was not only more likely, was also more likely to be statistically significant. And so, because we usually use small samples in humans and dogs, one potential concern that positive associations are accidents caused by measurement error is something that we can rule out. So we have some certainty in the reliability of this observation. Now, sleep spindle density, importantly, is not only associated with the animal's learning ability, but it also changes with age. And this is something which is, follows a bit more complicated pattern, in that what we know from humans, that sleep spindle density declines with age, we've only been able to demonstrate for slow spindles that we record over a centrally placed electrode. Whereas we also observe that fast sleep spindles recorded over the frontal electrode, where they are usually less abundant, increase with age in the dog. Or albeit we observe more fast spindles in the oldest dogs in our sample. This is quite unique to the dog. Only a similar effect is known for humans only from childhood to adolescence, where fast spindles increase as well in numbers. But I will speculate about the meaning of all of the results ultimately in the discussion. For now, as I promised you, we will also take a look at sleep spindle frequency, as several human studies had also shown that spindle frequency is one of the few characteristics that actually we do expect to increase with age. Unlike occurrence or the amplitude, that is the size of the events, spindle frequency, so the waves per second in a spindle event, was found to be higher in older humans. And this is what we also observed already in 2019 for older dogs. In old dogs, fast spindles over the central electrode displayed significantly higher frequencies. A year later, we were furthermore able to demonstrate that increase in spindle frequency between 2 measurements that were at least 3 months apart, so increase in spindle frequency between these 2 measurements, was associated with an increase in repetition errors on a reversal learning task, which was also tested at least 3 months apart. And this was also true for sleep spindles recorded over the central electrode, however, for both the slow and the fast subtype. What do different aspects of our results mean for the utility of the sleep spindle in dogs as a marker of cognitive aging? Well, first of all, something I didn't mention in the introduction to keep it as short as I can, but which is a potential concern in studying sleep spindles, is that there are other similar oscillations in the EEG of humans and other animals. And so it is very important to point out that an association between sleep spindle occurrence and learning was demonstrated to specifically concern the so-called thalamic spindle, which is what we consider to be the real spindle. That was demonstrated in an optogenetic experiment in mice, that this is a specific real spindle effect. And so the fact that we could observe learning spindle associations in the dog that are similar to the ones observed in humans and mice was a very important validation for our work in the dog model. Furthermore, our experiences in the dog, compared with some other observations that have accumulated in the last years across animals, are slowly building up to dispel a notion that was popular at least back in 2000 and 2005, where many researchers believed that we should expect spindle frequency to be very different between different species. But that's not what we see in the dog concerning events that we see are associated with cognition and aging similarly as in humans, as these events we detected with a frequency definition that follows the definition of human spindles. And combined with other animal work, we can conclude that sleep spindle frequency in general is a very conserved feature of the mammalian sleep spindle. And finally, one of our most recent findings in the dog concerning this association between reversal learning, repetition errors, and spindle frequency was excitingly discovered almost at the same time in humans. And this is crucial because spindle frequency and its relationship to cognition and aging is a bit tricky, in that in younger subjects, spindle frequency is actually positively associated with many aspects of cognition. And even though we have seen in humans spindle frequency increase with age and even be a good predictor of the onset of dementia, a specific cognitive correlate for spindle frequency was not discovered until 2020, when we showed it in the dogs and Guardani and colleagues showed it in humans. One possible explanation for this mixed picture on spindle frequency, and I try to keep it short, is simply that it might be a compensation effect that we know also from some other neural systems. As cells in some brain structures die, the firing frequency of the surviving cells increase, and so we can observe higher frequencies. And it seems that repetition errors in reversal learning are a type of mistake that the compensation mechanism cannot compensate for. And this is why this specific observation correlates positively with spindle frequency and other cognitive aspects do not. Thank you very much for your attention. I would like to thank my colleagues and all the dog owners and dogs which participated in the studies. If any of them are present in the audience, in particular, I know that it is not an easy type of experiment, and the anecdotes are very sticky, so I'm very grateful for everyone who stood with us. As you can see, we had some relevant discoveries based on this work. Thank you very much again. That's all from me.
Thank you very much, Ivo. That's very exciting to see dogs' behavior and the connection of what is happening in their brains. Thank you very much. I have a question. Do you know something about this specific brain activity marker and gender difference?
We actually did some work on that as well. I mean, it was left out from today's talk because of space and time issues. But yes, sleep spindles are actually among those brain oscillations that are sexually dimorph. And both in dogs and in humans, this has been demonstrated directly. And well, we're not entirely sure yet about what this exactly means in detail for cognition. We know that the spindles are different. In humans, we have some more direct links to cognition, because in humans it seems that only women profit from sleep spindles when it comes to intelligence. Intelligence specifically, we're not talking about sleep-dependent learning. And men do not. So male intelligence seems to be independent of sleep spindle expression. So it seems that the same construct, intelligence, is achieved through different strategies in men and women. But in dogs, we only could replicate some effects which are known and concern the general expression of sleep spindles. That is, fast sleep spindle densities and amplitudes and even frequencies were found to be higher in intact female dogs, and they tend to be highest at the peak of the progesterone phase of the menstrual cycle in women as well. So yeah, that will be regarding this question.
Great. So we have more studies forward. Very exciting. We have a question from Amanda. Can we influence the spindle frequency and so cognitive capacity? I think she's talking about training possibly. Well, please.
Well, one thing that people have been doing in the human and even in the rodent literature on sleep spindles is they have tried to stimulate the brain to produce more sleep spindles. And interestingly, this is something that the brain seems to be doing when either it is stimulated with a frequency that is similar to the frequency of a spindle. And so this has been done both optogenetically and with electromagnetic stimulation. And then the brain responds by producing more spindles. And that in turn has been shown to affect learning success in the mouse. This is this particular experiment which also demonstrated that it is the thalamic spindle and not any other similar oscillation that is connected to learning. But so since the question was about frequency, short answer is I don't know. What I've just said is particularly about occurrence of sleep spindles. But in order to increase the occurrence of sleep spindles, artificial stimulation needs to be in their frequency. So that is an important thing to note here.
Okay, so just a quick answer for this very last question. Was your study longitudinal? Was your dropout rate very high for dog EEG?
No, the study was not longitudinal. Well, actually, one of our studies was longitudinal. So I actually presented a combination of studies that we did in the last 4.5 years. And the one longitudinal study we had was the one demonstrating association between changes in frequency, so how many waves per second within a spindle, and the performance on the reversal learning task. So this effect was based on a longitudinal study. And yeah, we had dropout there for sure, part of which, however, was also not just because of owners and dogs not showing up. I mean, in that regard, we were actually rather lucky. We have a good relationship with owners and dogs, but sometimes some of the data files, unfortunately, were corrupted. We found out before analysis, and well, a lot of things can happen, of course. We have to say those particular results were based on what remained in the end. Yes.
Okay, so thank you very much, Ivo.